Hello, my name is Sashel King, and today we will be presenting genetics of populations. So, on this slide, we will talk about genetic structure of populations. So, we'll dive deeper into gene flow and genetic drift in our slides to come. We've talked about natural selection before, so let's do a quick overview again. So, natural selection favors certain alleles based on their fitness in particular environments. It can lead to adaptation and divergence among populations as they adapt to different ecological niches. We've also talked about mutations before. So, mutations introduce new genetic variation into populations, where mutations can become more common over time if they confer a selective advantage. Now let's move back up to number one and number two. So, gene flow. Gene flow involves the movement of individuals and their genes between populations. High gene flow tends to combine populations genetically, while low gene flow usually leads to more genetic differentiation. And then number two, genetic drift. So, genetic drift refers to random changes in allele frequencies in a small population. It can also result in the fixation or loss of alleles and increased genetic divergence among populations. Okay, so let's deep dive into gene flow, also known as migration. So let's talk about some of the molecular markers. DNA markers such as microsatellites, SNPs, and DNA sequences are commonly used to assess genetic diversity and structure. Genetic data are analyzed using various statistical methods, including F-statistics, principal component analysis, PCA, and clustering algorithms. Okay, so let's move into our applications. So, conservation of genetics. Understanding genetic structure helps identify populations at risk of inbreeding and guides conservation efforts to maintain genetic diversity. Number two, phylogeography. Genetic structure studies contribute to our understanding of historical biogeography and population migration patterns. And number three, forensic genetics. Genetic structure analysis is used in forensics for human identification and tracing the origins of biological samples. The genetic structure of populations is a dynamic and complex field with broad implications for biology, ecology, and conservation. Molecular tools and analytic methods have revolutionized our ability to uncover the genetic intricacies within and among populations. So let's talk about another structure. We'll talk about genetic drift. Genetic drift and gene flow are two fundamental processes in population genetics that influence the genetic makeup of these populations. They both play significant roles in shaping the genetic diversity within and among populations. Genetic drift is a random process that affects allele frequencies within a population over time. It occurs due to chance events, particularly in small populations. So let's talk about some key points to understanding genetic drift. So the first one we'll talk about will be random sampling. In a population, individuals reproduce and pass on their genetic material to the next generation. However, not all individuals reproduce, and the genetic material passed on is a random sample of the parent generation genes. Let's talk about effects on small population sizes. Genetic drift has a more pronounced impact on small populations because there are fewer individuals contributing to the next generation's gene pool. In larger populations, the effects of genetic drift are much weaker. Okay, let's talk about genetic variation and the loss of it. So, over generations, genetic drift can lead to the fixation of certain alleles. All individuals in the population carry the same allele, and the loss of others. This reduces genetic diversity within the population as well. So let's talk about the founder effect and the bottleneck effect. So the founder effect is when genetic drift is especially evident in situations where a small number of individuals establish a new population. The genetic makeup of the founder population may not accurately represent the genetic diversity of the source population, leading to a founder effect. So let's move on to the bottleneck effect. A population bottleneck occurs when a significant portion of a population is suddenly reduced in size due to a catastrophic event or other factors. Genetic drift during a bottleneck can result in a loss of genetic diversity as well. Gene flow, also known as migration, is the movement of individuals and their genetic material, alleles, between populations. Let's go over some key points to understanding gene flow. So first we have an introduction of new alleles. Gene flow introduces new alleles into a population when individuals from one population migrate and reproduce in another population. This can increase genetic diversity within a population. Next, let's talk about homogenizing effects. So, gene flow tends to reduce genetic differences between populations. Populations with high rates of gene flow are genetically more similar to each other. Now let's talk about adaptive potential. Gene flow can bring beneficial alleles into populations, potentially enhancing their adaptive potential. It can help populations adapt to changing environments by providing new genetic variation. Next, let's talk about barriers to gene flow. There's physical barriers like mountains and rivers, and ecological factors, differences in habitat preferences, that can restrict gene flow between populations. Populations that are isolated by such barriers tend to diverge genetically. Lastly, let's touch on hybridization. In cases of significant gene flow between populations, hybridization can occur. This can lead to the creation of hybrid individuals with a mixture of alleles from both populations. So to summarize genetic drift and gene flow. Genetic drift is a random process that can lead to allele frequency changes in populations, especially in small populations, and it tends to reduce genetic diversity. On the other hand, gene flow is the movement of genetic material between populations, which can increase genetic diversity within populations and reduce genetic differentiation between populations. Both processes, along with natural selection, mutation, and other factors, contribute to the genetic structure and evolution of populations. So let's talk about the Hardy-Weinberg law. The Hardy-Weinberg law, also known as the Hardy-Weinberg equilibrium, is a fundamental principle in population genetics that describes the distribution of genetic variation in a population under specific conditions. It was developed independently by G. H. Hardy and Wilhelm Weinberg in the early 20th century. The Hardy-Weinberg law provides a baseline against which one can assess whether evolutionary forces are acting on a population. So let's talk about some of the key aspects of the Hardy-Weinberg law. The Hardy-Weinberg law serves as a null hypothesis against which observed genetic data in real populations can be compared. If the observed genotype frequencies deviate significantly from those predicted by the law, it suggests that one or more of the conditions for genetic equilibrium are not met. Deviations from Hardy-Weinberg equilibrium can indicate the presence of evolutionary forces such as genetic drift, natural selection, mutation, migrations, or non-random mating in a population. The Hardy-Weinberg equilibrium is a fundamental concept in population genetics and is used in various fields including evolutionary biology, medical genetics, and forensics. In practice, most natural populations do not precisely follow the Hardy-Weinberg equilibrium due to the influence of various evolutionary forces. Deviations from the HWE provide valuable insights into the mechanisms driving genetic change and evolution within populations. So let's talk more about the conditions for the Hardy-Weinberg equilibrium. So number one would be random mating. Individuals in the population mate randomly with respect to their genotype. This means that there is no preferential mating based on genotype. Number 2 would be a large population size. The population is sufficiently large so that genetic drift is negligible. In small populations, genetic drift can have a significant impact. Number 3 would be no mutations. There is no introduction of new alleles into the population through mutation. Number 4 would be no migration. The population is closed, meaning there is no migration of individuals in and out of the population. And lastly, we have number 5, no natural selection. There is no differential survival or reproduction based on genotype. All genotypes have equal fitness. The Hardy-Weinberg law is often expressed using mathematical equations. If we denote the frequency of one allele as lowercase p and the frequency of the other allele as lowercase q in a diploid population where individuals have 2 alleles each, the law states the frequency of a homozygous individual for allele p is given by p squared. The frequency for a homozygous individual for allele q is given by q squared. The frequency of heterozygous individuals, genotypes PQ or QP, is given by 2PQ. The sum of these genotype frequencies equals 1 or 100%. So we have P squared, frequency of homozygous dominant, plus 2PQ, frequency of heterozygous genotypes, plus Q squared, frequency of homozygous recessive genotypes equals 1. You can also see a clearer picture in the photos that I have provided. So now let's move on to genetic variation. So let's ask ourselves, what is genetic variation? Genetic variation refers to the differences in DNA sequences or genetic markers among individuals within a population or species. This variation arises due to mutations, genetic recombination during reproduction, and other processes. So let's talk about genetic variation in space. So genetic variation in space refers to how genetic diversity varies across different geographic locations or regions. Populations that are geographically isolated from each other often exhibit genetic differentiation. This occurs because of limited gene flow between these populations. Geographic barriers such as mountain, rivers, or oceans can restrict gene flow and contribute to genetic differentiation. Populations in a different environment may also experience divergent natural selection, leading to the development of unique genetic adaptations. So let's talk about genetic variation in time. So in time refers to changes in genetic diversity over generations. Populations can evolve over time due to various factors, including mutation, natural selection, genetic drift, and migration. Over long periods, new genetic variants may arise through mutation and become more or less common in a population. The genetic composition of a population can change in response to environmental changes or selective pressures. Maintenance of genetic variations could be things like genetic diversity within a population, which is essential for its ability to adapt to changing environments. Genetic variation can be maintained through mechanisms such as balanced polymorphism, frequency-dependent selection, and spatial heterogeneity in selection pressures. So let's talk about the importance of genetic variation. Understanding genetic variation in space and time is crucial for various fields, including evolutionary biology, conservation of biology, and human genetics. It helps scientists track the evolutionary history of species, assesses the impact of environmental changes on genetic diversity, and identifies populations that may be at risk of genetic depletion. To summarize, genetic variation in space and time is a dynamic and complex topic that addresses the distribution and changes in genetic diversity within populations and species. It plays a central role in the study of evolution, population genetics, and the conservation of biodiversity. So let's talk about genetic forces. Gene frequencies within a population can change over time due to several evolutionary forces and processes. These forces are fundamental in shaping the genetic makeup of populations. So let's talk about some of the primary forces that change gene frequencies. So number one would be natural selection. Natural selection is defined as the process by which certain alleles become more or less common in a population based on their impact on an organism's fitness or given environment. For example, we have the peppered moths. So in the 19th and earliest 20th centuries, peppered moths in England existed in two color morphs, light and dark. During the Industrial Revolution, population darkened tree bark, making dark-colored moths less visible to predators. Natural selection favored the dark-colored moths as they were better camouflaged against the darkened tree trunks. Over time, the frequency of dark-colored moths increased while the light-colored moths declined. So let's move on to genetic drift. So we've already spoken about what genetic drift is. So let me give you an example. So let's talk about the founder effect in Pinalab Atoll. In Pinalab Atoll, a small island in the Pacific, a typhoon reduced the population to only a few survivors. These survivors carried a unique allele for color blindness due to their isolation. So in this small population, genetic drift resulted in a high frequency of color blindness. So let's continue with genetic forces. So an example of a mutation as a genetic force could be seen in sickle cell anemia. Sickle cell anemia is caused by a mutation in the HBB gene, leading to the production of abnormal hemoglobin. In regions with a high prevalence of malaria, individuals carry one copy of the sickle cell allele, have a survival advantage against malaria. As a result, the frequency of sickle cell allele is higher in malaria endemic areas. Okay, let's move on to gene flow, which we've already spoken about. So an example for gene flow can be seen in bird species distribution. Consider two isolated populations of a bird species, one with bright blue feathers and another with dull brown feathers. If some individuals migrate and mate between these populations, gene flow can result in intermediate offspring with mixed feather colors. And so let's move on to number 3, non-random mating. So an example for this could be positive assortative mating in humans. In some human populations, individuals with similar traits such as height or education level are more likely to marry each other. This can lead to an increase in homozygosity for certain alleles related to those traits within the population. And let's talk about another example with the bottleneck and founder effect. So for the bottleneck effect, we can see this in northern elephant seals. Northern elephant seals were heavily hunted, resulting in a severe population bottleneck in the 19th century. Today's population is descended from a small number of survivors, resulting in a low genetic diversity.